guys, welcome back to my Andrew Space. Hope you all are doing and learning well. In this video, we are going to look at the structure and differences among the chromatin, chromosomes and chromatids. Many of you get confused by these three terms. Some thought that they are synonymous. So this video will clear all your doubts regarding chromatin, chromosomes and chromatids. This video is further in continuation with the series of nucleus as chromatins, or we can say chromosomes, are one of the most important component of nucleus. So without further ado, let's get started. What are the chromatins? Basically, this word chromatin is taken from the Greek word chroma, okay, which means color. The chroma in Greek means color, okay. So these are thread-like structures that absorb certain dyes, thereby looks colored. Now look at this picture. Look at this picture. You can see there are lots of thread-like structures. Some are green, okay. Some are light yellow, dark green, red, purple. These all thread-like structures are present inside the nucleus. Okay. So these thread-like structures are chromatin. These are chromatins. These are basically complex of DNA, RNA, and proteins. These chromatin structures become more thick and compact and form chromosomes during cell division. Okay, this thread-like structure, suppose this is our nucleus and there are thread-like structures. These are chromatins. There are lots of chromatins inside the nucleus. Okay, but during cell division, in the process of mitosis and meiosis, these get compact and form structures like this. Okay, now this time these are known as chromosomes. We also know that DNA is having double helical structure and also known as genetic material, but DNA does not appear as free linear strands in the nucleus because it is too long to fit inside the nucleus. So why these thread-like structures get compacted? Because they have to fit in this particular space of nucleus. Okay, DNA is in highly condensed form and wrapped around nuclear proteins in order to fit inside the nucleus. So these DNA get compacted with the help of the proteins that are present inside the nucleus, and these proteins are basically histone proteins. So DNA get condensed and form wrapped around nuclear proteins, histone proteins, so that they can fit inside the certain space that is present inside the nucleus. So we can say about chromatin that these structures are made up of DNA, RNA and proteins. Those proteins are known as histone proteins. Okay. It condenses during cell division or in the process of mitosis and meiosis and becomes a chromosome. Hope you understand that suppose this is our nucleus and there are so many thread-like structures inside the nucleus. This time they are known as chromatin. Okay, they are chromatin. But during cell division, they are again get compacted and become dense structures. Okay, so they become like this. And so chromosomes are much condensed structure than that of chromatin. Chromosomes are unwound, condensed structures, whereas chromosomes are highly packaged and more condensed than that of chromatins. Okay, so as I just told you that chromatins are thread-like structures, but if you see under the microscope, it looks like dotted. Okay, if we see this particular, let me magnify this particular chromatin. If we see this structure under microscope, you will see there is thread and over which some dotted like structures are present all over the string. Okay, let me change the color. Okay, we will take red. So under microscope, you will see the thread of chromatin will look like this dotted structures. Okay, so at its extended form, means loosely compact form, chromatin looks like beads on a string. The beads are called nucleosome. So after magnification, you will see the chromatin structures look like a string in which there are some dots all over the string. So this particular dot is what? This is our nucleosome. Okay, this is nucleosome. So let's discuss what is the nucleosome. Now, what is nucleosome? So nucleosome basically acts or play a role of beads on a string model of chromatin. The chromatin having a model beads on a string in which the bead part is played by nucleosome. So as I just told you in the previous slide that these dotted like structures which are arranged linearly on a string. So this bead is what? This is our nucleosome. You can see this structure, okay? These parts, first, second, third, fourth, fifth, and sixth. They looks like dot, okay? They looks like dots. This particular portion is bead on a string and these beads are known as nucleosome. So it is the structural unit of DNA packaging in eukaryotes and also known as fundamental subunit of chromatin. Basically, nucleosome is a complex structure which is made up of DNA and histone proteins. Okay, this particular chromatin is a complex which is made up of DNA, RNA and histone proteins. It is the segment of DNA wound around 8 histone proteins and looks like thread wrapped around a spool. Okay, so what it says that this is our DNA. Okay, how this nucleosome structure is formed? Basically, it is formed when the DNA is wrapped on 8 histone proteins. Suppose, let me draw. We have four histone proteins. The first one is H2A, the second one is H2B, the third histone protein is H3, and fourth is H4. Okay, so these are four histone proteins that will ultimately form octamer. Exact four histone proteins copied and they become four. They become again four. So two copies of each of the histone proteins form this histone octamer. Two histone proteins of H2A, two histone proteins of H2B, two histone proteins of H3, and two histone protein of H4. They form a complex structure which is known as histone octamer. This is histone octamer. Okay. So the DNA is wrapped around this histone octamer, which looks like thread wrapped around a spool. Okay. So less than 2 terms of DNA wrapped around a set of 8 histone proteins. Just look. This is our DNA. Okay. Let me change the color. Okay. So this is our DNA. It will not take 2 turns. Okay. In fact, less than 2 turns of DNA over the nucleosome. Hope you understand this structure. And this is the magnified image of this DNA. Molecularly, the nucleosome core particle consists of 146 base pair of DNA wrapped in 1.67 left-handed supercoiled superhelical turn around a histone octamer. Hope you all get understood the nucleosome structure, which resembles bead in a chromatin. If you still not get my point, let me explain you once again. Suppose this is H2A, histone protein H2A. We have two exact copies of H2A. Okay. Then we have H2B, two exact copies of H2B. Now, third, we have H3, two copies of H3 and two copies of H4. Okay. Next are DNA. So this particular core is what? This is our histone octamer. This is our histone octamer because it consists of 8 histone proteins. And this particular nucleosome consists of 146 base pair of DNA. So what stretch of DNA is wrapped over this nucleosome? These are 146 base pair of DNA. And they are not completely take 2 turn. The DNA does not completely take 2 turn. In fact, less than 2 turn, that is 1.67 left-handed superhelical turns. Okay, so this is our DNA. It will take 1 turn. Okay, and then second turn. And then it again join with another nucleosome, like so on. Okay. So each core particle of histone are connected together by stretches of linker DNA, which is about 80 base pair long. The first nucleosome, this is connected to the second nucleosome. This is our first nucleosome and this is our second nucleosome. Okay. So they both are connected together with some stretch of DNA, which is about 80 base pair long. This is 80 base pair long. And this particular DNA which connected two nucleosomes together is known as linker DNA. Okay, this particular is our linker DNA. So 80 base pair linker DNA. This is our linker DNA. Now, what is linker DNA? So, as I just told you, in the model of beads in a string of chromatin, the bead part is nucleosome. Okay, this bead part is nucleosome, and this string which connected nucleosomes all together, this string part is played by our linker DNA. So this string part is our linker DNA. Okay, this is our linker DNA. Now take a look at this diagram. Take a look at this diagram. So there are 8 histone proteins. The first one, this one, second, third, fourth, and this is fifth, sixth, seventh, and eighth is just behind this. Okay, so they are forming octamer having 2 copies of each H2A, H2B, H3 and H4 histone, and this complete make histone octamer. Okay, it will make histone octamer. This red color string is our DNA that wrapped on histone octamer. This one, this red, this is our DNA that's wrapped on the nucleosome. Okay, this particular DNA that makes less than 2 turn on the nucleosome, and beside this you can see there are some purple DNA also. This purple DNA is known as linker DNA because it helps in linking 2 nucleosomes all together. Okay, so linker DNA is double-stranded DNA of about 38 to 53 base pair long in between 2 nucleosome core that in association with histone proteins H1 holds the cores together. I will describe you this H1 histone proteins in a short while. Okay, so this is basically linker DNA. As its name suggested, the linker DNA is basically responsible for linking 2 nucleosomes. Okay, so again, linker DNA acts as a string in beads in a string model of chromatin. So now, what is chromatosome? This is quite new term for you, I think. So these are chromatosomes. What are chromatosomes? Basically, chromatosome is a complex of nucleosome, okay, nucleosome plus another histone proteins, that is H1 histone protein, okay, H1 histone protein. If this complex is formed, then this is chromatosome. We are not considering linker DNA in this. We are just taking nucleosome and H1 histone protein. So the complex of nucleosome and H1 histone protein is what? It is chromatosome. So this structure is formed when histone 1 binding to a nucleosome. So this is our nucleosome, okay, and this green color, this one, this green color protein is histone H1 protein. It also plays the role of linking up, okay, linking up of two nucleosomes. The chromatosome contains 166 base pair of DNA, 146 base pair from DNA wrapped around histone octamer, and 20 base pair are from the histone H1 binding to the nucleosome. So, suppose we know that the nucleosome having a core DNA that is of 146 base pair. This red color is 146 base pair DNA, okay, and the other 20 base pair are on this histone H1 proteins, okay. This is what? This is 20 base pair. So the chromatosome having a complete stretch of DNA is about 166 base pair. Sorry, it is 166 base pair. 146 from the nucleosome and 20 base pair from this histone H1 protein. Histone H1 is a linker histone, and protruding the DNA stretch from the linker histone is known as the linker DNA. So the stretch that is arising from this H1 histone protein, this is our linker DNA, okay. So this particular structure having nucleosome and the H1 histone protein, which includes the 166 base pair of DNA stretch, is our chromatosome, okay. And from this chromatosome structure, the some part of the DNA stretch that comes out from this H1 histone protein is our linker DNA. Now, forms of chromatin. Basically, chromatin exists in two forms. Okay, the first one is euchromatin and the another one is heterochromatin. Euchromatin has loose chromatin structure and active for transcription, whereas heterochromatin has condensed chromatin structure and is inactive for transcription. So in this diagram, you can see here that this is our chromosome, okay. This is our chromosome having arms known as this. The center one is centromere, where these two chromatids get attached, okay. So if you can see at the telomeric end, okay, at the telomeric end and at the centromere region, the DNA is very tightly packed, okay. So highly condensed structure of chromatin is found at the centromeric region and at the telomeric region. These are telomeres. These are telomeres. So these highly condensed or compact chromatin forms heterochromatin, which are present at the telomeric and the centromeric, okay, and at the distal ends of this strand of this particular region is occupied by loosely, or we can say lightly packed chromatin. So these are euchromatin. Okay, these are euchromatin. Heterochromatin, the genes which are present in this region, as they are very tightly packed, so they are inaccessible to the polymerase enzyme. So there is no transcription takes place. No transcription takes place at the heterochromatin. On the other hand, these chromatin are lightly packed. Okay, so they are accessible to the polymerase enzyme. So the transcription is happened in the euchromatin region. Let's discuss more about the euchromatin and heterochromatin. So as we know that chromatin get condensed and attain the shape of chromosomes. Okay, so let me explain you with the diagram. This is our chromatin, okay, which are compact structure and form what? They form chromosome. These are nothing, just a compact version of chromatin. And this structure is formed during the mitosis and meiosis process, okay, and they are attached together at the centromere. Okay, so some regions are tightly compact. Okay, some regions are tightly compact, tightly packaging are present at some portion, and some portion are loosely packed. This region, this particular region of chromosome is loosely packed. Okay, so euchromatin is lightly packed or decondensed form of chromatin, which is usually found at the distal end of chromosomes. These are present at this ends of chromosomes, or we can say on the arms of chromosomes. It is usually dispersed all around the nucleus and is replicated throughout the S phase. Okay, so let me make you understand. Suppose this is our nucleus and this euchromatin is present at this region of the nucleus, at the center region of the nucleus, whereas the heterochromatin, which is tightly compact structure of chromatin, the heterochromatin is present at the periphery of the nucleus. Okay, this is present at the periphery of the nucleus. So this is our euchromatin. This is our euchromatin. So it is dispersed all around the nucleus and is replicated throughout the S phase. During synthesis phase, this euchromatin region get replicated. Okay. This region of DNA is enriched in genes that are often actively transcribed, that is turned on genes. So the genes that are present in euchromatin are known as turned on genes because they are actively transcribed and they are closely associated with RNA polymerase, as this structure is loosely compact and loosely lightly packed, so they are easily accessible for the polymerase enzymes. Thereby euchromatin is transcriptionally active form of chromatin. So the euchromatin, this 92% of the human genome is euchromatic genome. This 90% of the human genome is euchromatic. That means 90%, 92% of the human genome is actively transcribed. Okay. Next. Heterochromatin. On the contrary to the euchromatin, the heterochromatin is tightly packed form of DNA or condensed DNA that is typically found at the centromere and telomere. So, if this is our chromosome, okay, this is our chromosome, the area that the chromatin that is present at the centromeric region and that are present at the telomeric end of chromosome, okay, sorry. These are tightly packed. These are tightly packed, or we can say dense chromatin are found at these regions. These are heterochromatin. So these are present at the centromeric and telomeric region. Because it is tightly packed, it was thought to be inaccessible to polymerases and therefore having inactive genes, that is turned off genes. Okay, so if something is very compact in structure, so the polymerase could not make it way to come and replicate it or transcribe it. So these particular regions are known as transcriptionally inactive. The genes that are belongs to these regions are inactive genes. Okay, or we can say turned off genes. Therefore, it is transcriptionally inactive form of chromatin. It mostly consists of repetitive DNA sequences and is relatively gene poor. Okay, these regions are gene poor, whereas the regions that is present in the euchromatin area are gene rich. So heterochromatin are gene poor, whereas the euchromatins are gene rich. These are gene rich region. It is mostly notable property that it is ability to silence euchromatic gene expression. So the property of these heterochromatin is in silencing the gene expression of these euchromatic genes. Okay. Next. Now, what are chromosomes? Actually, chromosomes are nothing but the more compact version of chromatins. So, chromosomes are made up from chromatin, in which the chromatins are arranged in more compact version. Okay. So, suppose if this is our chromatin, if it get more condensed structure, okay, during the process of cell division, it compacted again and again and formed this type of structure. This particular is our chromosome. Okay. So the chromatin condenses to form chromosome during cell division, that is mitosis or meiosis process. But why chromatin further wraps? As it was wrapped during nucleosome wrapping. So why it is necessary to wrap again? The answer is, beside nucleosome wrapping, the DNA must be compacted again to fit inside the nucleus. Therefore, chromatin further compacted by a series of more complex structure known as chromosome. Okay. So this is also a chromosome, this one, and this one is also a chromosome. But in this, there is only one chromatid. Okay. This is only one chromatid. But during duplication, okay, during duplication, or we can say replication, the two chromatids formed, okay, this is one chromatid and this one is another chromatid. The two chromatid form, which are connected together by a centromeric region. This one is centromere. Okay. In human cell, each cell normally contains 23 pairs of chromosomes and a total of 46. So we have 23 pairs of chromosomes. Now, what are chromatids? So you might confused with these two terms, the chromosomes and chromatids. Let me explain you. Suppose this is our cell, okay, and having chromatins. These are chromatins, okay. These are our chromatins. During cell division, these chromatins get compacted again and form structure like this, which is much compacted version. Okay. This is much compacted version of chromatins. So now these are known as chromosomes. These structures are now called as chromosomes. These are chromosomes. They are having, this time they are having only one chromatids. They are having one chromatids. This complete region, okay, this complete region is known as chromatids, having only one chromatid. And after duplication of the DNA, they become like this. This particular area get duplicated and this particular chromatid get duplicated. So they become like this, one chromatid and two chromatid that are joined together with a centromere. Okay. And same like this particular is duplicated, which are connected all together with the centromere region. You can see in this diagram, this is our chromosome, okay, having a single chromatid. this is single chromatid. Okay, and when replication occurs, this duplicated and form two chromatids. These are two chromatids which are joined together with the centromere. Now these two chromatids are known as sister chromatids. Okay, so a chromatid is one half of a duplicate chromosome. Here what we can say, we can also say that this is a single chromatid, or we can say that after duplication the half part, okay, this half part is a chromatid. Before replication, one chromosome is composed of one DNA molecule. In replication, the DNA molecule is copied and the two molecules are known as chromatids. Okay, this is one molecule, and after replication, they double up and become two, and now these two chromatids are what? They are sister chromatids. Thanks for watching, and for more such interesting videos, stay tuned with us.